Proteins are the future of medicine as they can be used to treat various afflictions like diabetes, infectious diseases, and different forms of cancers. To create protein-based therapies, we start by making lots and lots of versions of a protein and then seeing which ones can bind or hug a troublesome target tightly. Why is hugging tightly the goal? Well, imagine someone getting tackled in football. The proteins we create kind of the same thing where if they can tackle a troublesome target, it could be slowed or stopped and be therapeutic for the patient. To think about undruggable on a cellular scale, some of the targets on the cell surface cannot be targeted and are termed undruggable. Think of undruggable as being unable to tackle the #1 football player in the nation. The cell surface has a lot of different structures on it, and similar to the players, we try to target that target itself. Methods to do this are working on a case-by-case basis while also being time-consuming, complex, and/or expensive. This is problematic as the cell surface is a hot target when dealing with things like multi-drug resistant bacteria, nasty viruses like the ones that cause COVID-19, and even us. So what if I told y'all that I'm working on a technology that bypasses the need to isolate this target? I plan on using the cell itself to help me engineer medicine. To create these tackling protein drugs, I will start by making lots and lots of protein. Each protein has a DNA tag attached. The role of the DNA tag is to serve as a label that reveals the protein's identity. I will also attach DNA to the cell surface target of interest. The key is that I'll modify both the DNA attached to the cell surface target and the proteins in a way that makes them perfect for each other once they meet. Romantic, right? Once there's interaction, a unique structure forms and produces a signal telling us that this is a protein that tackles our target and we want to keep it, while proteins that tackle the cell but not the target won't produce that signal. So far, I have the DNA tags, the unique structure, and the signal all figured out, but these proof of concept experiments are done without the proteins and cells. I plan on getting much closer, however, by attaching DNA tags to several proteins and then seeing which ones can produce a signal from tackling that target while facing competitions from others that don't. In the end, my technology will allow for proteins to be the go-to when dealing with undruggable targets on the cell surface. As more drugs are made and tested, the chances of one being approved increases. And in the end, these very special proteins will help us tackle human diseases relating to undruggable targets. Talk about a game changer.